Thanks, Olivier, and thank you to all the organizers for having me today. So as Olivier mentioned, I work in the School of Medicine at UCSD in Jeff Eska's lab. And so today I kind of wanted to give you a lot of experimental details on how we utilize genome-wide CRISPR screening to study the biosynthesis of a really important biomolecule, heparan sulfate. And so just a little bit of background on heparan sulfate. It's highly sulfated, long linear polysaccharide chains that are attached to core proteins on the surface of virtually all animal cells. And these heparan sulfate chains are very heterogeneous, and they consist of alternating sequences of disaccharide subunits. And these sugar units are highly sulfated at specific positions, shown here in these 2S, NS. There's different sulfation sites along the heparan sulfate chains. And what these sites do is they create binding domains for proteins such as growth factors or coagulation factors like antithrombin. And as a result, these heparan sulfate chains are involved in a lot of important cellular processes such as proliferation, motility, development, coagulation, and also angiogenesis. And while it's involved in a lot of important processes, it's also been implicated in its role in certain diseases, such as cancer or neurodegenerative diseases like Alzheimer's, and also infection. Many viruses bind to heparan sulfate on the cell surface, and that allows them to eventually get in and infect the cell, and also inflammation. And the biosynthesis of heparan sulfate mainly occurs in the ER and the Golgi of the cell. And there's many enzymes, all shown here, that are instrumental in constructing these long polysaccharide chains and modifying them, such as sulfating them in the Golgi. But what's not really known right now is are all these enzymes and how is the biosynthesis of heparan sulfate regulated? And so you can imagine there's a transcription that occurs, and then you get biosynthesis of these polysaccharides in the Golgi and the ER. Then they're transported to the cell surface, and they can be modified while they're on the cell surface in the extracellular matrix. Then they can be endocytosed and degraded or recycled back to the cell surface. So there's a lot of potential potential sites along the pathway where regulation of the biosynthesis or the presentation of these sugars on the cell surface. So the whole goal of my project was, can we use genome-wide screening to try to further understand the regulation of the biosynthesis of heparan sulfate? And there's a lot of downstream potential applications to this. For instance, finding novel drug targets to where you could potentially control the biosynthesis of these sugars in cancer cells or in other types of disease states. And so this is just showing that we could potentially, when we do these CRISPR genome-wide screens, the guide RNAs that are targeting different types of genes could fall into many different types of categories. And so in terms of this workshop, I wanted to give a little bit more detail on how I actually performed the screen from the very beginning in the application of trying to find novel factors that might be regulating heparan sulfate biosynthesis. And so we utilized the genome CRISPR screening using a positive selection. So I think the next presenter will talk maybe more about negative selection, where we're looking at dropout of genes. And what I'm looking for is enrichment of genes when you treat cells, for instance, with different types of toxins. And I'll give you some more details on that next. So up here at the top, we're just showing that we're using the Gecko version 2 library that was mentioned earlier today. And so this targets over 19,000 genes, and you have 6 guide RNAs per gene. So we just ordered these from Adgene, or there's other vendors that sell this library now, such as Agilent, who's here today. And this was designed in the Zhang lab at MIT and was published back in 2014. And so, as was mentioned in earlier talks, when you purchase this, you get a small amount of the plasmid library. So we first needed to amplify this in bacteria. And so after we were able to do that, we use HEC293 T cells to make our lentiviral library. And so after you do that, we can isolate the lentivirus, and we can transduce Cas9 expressing cells. And we do this at a very low MOI to ensure that each cell only gets one guide RNA or one viral particle. And we're using these A375 cells, which is a human melanoma line, really easy to grow. It's actually it was shown it was used with the Gecko version 2 library. And the only thing we had to do is express Cas9, and I'll give you some more details about that later. So as I mentioned, we transduce at a very low MOI, and then you select with purimycin to get rid of any cells that did not get a viral particle. As a result, you get this whole library of cells where each cell has a different gene that's been targeted with a different guide RNA. And then what's nice is now you can freeze down this library, and you can use it for whatever you like, if you want to do negative selection screens, or in our case, positive selection. And so in our case, we designed a screen using different toxins. And so shown here on the left, these are 3 different toxins that require binding to heparan sulfate on the cell surface in order to get into the cell and kill the cells. So if heparan sulfate is not present, or there's reduced amounts, it will reduce the amount of toxin that will be able to get into your cell and kill the cells. And on the right here, as a counter screen, we use 2 plant lectins. So these lectins bind to other types of sugars on the cell surface. And what's important for this is that we can compare the hits we get, or the enriched guide RNAs from these toxins versus our heparan sulfate dependent toxins. And we can look at the overlap between these 2 screens. And what that helps us do is it sorts genes that are specific for heparan sulfate biosynthesis, and not just general glycosylation pathways, for instance. And also we take a PBS control sample along, as Philip mentioned, where you treat with PBS and you keep those cells going along. And I'll show you how we do that. As a result, we collect these resistant cells that survive the toxin treatment of each of these, and we can extract the genomic DNA and use deep sequencing to sequence for the guide RNAs present in our cells. And it's a little bit more details about these toxins. FGF binds to heparan sulfate on the cell surface along with its receptor. Diphtheria toxin binds to HBEGF and then also heparan sulfate. And then GNEO is a small molecule that binds specifically to heparan sulfate and can shuttle cargo into cells. So for instance, Heparan, which is a toxin that doesn't get into cells by itself. And as an additional screen, we also used FACS sorting, and we use this to help us determine our most promising candidates from both of our screens. So I'll give you some more details about that. It's the same way we're collecting low binding cells versus normal binding. We can do the same thing and sequence them. So before we can transduce our library, as I mentioned before, we first need to make Cas9 expressing cells. So to do that, we use this lenti Cas9 blast plasmid. We make the lentivirus, transduce our cells, and you can use qPCR to look at Cas9 expression versus wild-type cells. And as JP mentioned earlier, you can also use Western blot as well. And so this is important, showing that, yes, Cas9 is expressed in our cells. And then also, the next thing we want to do is the CRISPR-Cas9 system working in our cell line? I think this is important to validate before you go through the massive number of plates that you're going to be doing with these screens. And so I don't think it was mentioned yet today, but this is a kit along with, if you've heard of the Surveyor kit. These are really nice mutation detection kits where you can just PCR amplify the target of your guide RNA, the targeted region of the genome. And this kit, you hybridize your DNA, and there's an enzyme that goes along, and when you hybridize wild type with your knockout cell line, you can look at if there's any mismatches or indels at your target locus. And if there are, you'll get cutting occurring in your PCR product, and you can monitor that, visualizing these cleavage products. So I highly recommend this kit versus the Surveyor kit. I think IDT makes that. This is a lot easier, and you can be done in like 2 hours. Surveyor takes a lot longer, and sometimes it doesn't even work as well. So I'm not being paid to say that. I just think this is a really nice kit. And if you're not using virus as well, you can literally scrape off of the plate with a tip of a pipette and PCR off the cells and do this as well. So if you're just transfecting the CRISPR system, just a little insight into that we found this to work really well. And so also what we can do is look at functional assays to see if you're indeed introducing a mutation at your target gene. For instance, this NDST1 is a gene involved in sulfating, heparan sulfate. If you don't have that, molecules like GNEO or diphtheria toxin will not bind to heparan sulfate on the cell surface. So you do toxin-based cell survival assays like cell titer blue and look at resistance to certain toxins. MGAT1 is involved in genital glycosylation pathway for WGA to bind to a specific glycan. If you knock this out, you see resistance to this. Or this HBEGF, which is the #1 cell surface target for diphtheria toxin, you also see resistance. So these are just control experiments before we go through, and now we can transduce our Cas9 expressing cells. You end up with this pool, and then you're ready to screen. And we use a very strong toxin selection to minimize the number of hits that we get. You can obviously use less toxin selection if you want to kind of open the window, so to speak, of the guide RNAs that you'll get in return. So this is the overview of our toxin-based screen where we treat our pool of cells that have our gecko library in them. For 4 days with toxin, you get massive amount of killing, and then you let the cells grow back a bit just so you don't lose any guide RNAs when you go to isolate the genomic DNA and all the downstream applications. And then in the meantime, we also take PBS, and as you can imagine, these cells are continually growing, so you need to passage them while also maintaining high coverage as well. For instance, 100-fold coverage, which is what we did in this experiment. And giving some details on how we process our genomic DNA. So once you have your surviving cell population after toxin treatment or in your PBS control, you need to extract the genomic DNA. We use this QIAGEN kit. And the important part is we load all of the genomic DNA from all of your samples into PCR1 reactions. And so you can imagine this can be a lot of PCRs, as mentioned before, but we had to optimize this, and the most that we could get in was 2 micrograms of genomic DNA per reaction. And we use the Q5 high-fidelity polymerase. You want to use a high-fidelity polymerase because you're going to be sequencing these amplicons. And we used 18 cycles, and we used primers from the original Gecko paper. I know other primers have been implemented through Kristen and the IgM core. So we used these primers that would amplify the guide RNA locus in all of our cells, because remember that the guide RNA has been stably integrated into the genome with the lentivirus. So then for PCR2, you take 5 microliters of PCR1 and do 24 cycles. These are some examples of what we use for our PCRs. We have the barcode here in red, and then you have to introduce these staggered sequences in order for the sequencer to see your library as diverse. We had to troubleshoot that quite a bit. Kristen helped us a lot with that. And so you get a gel that looks like this. Again, we don't run a gel until after PCR2. And then what we do is we cut the gel here and do a gel extraction, and then just rerun to ensure that you just have a single band. And then we can quantify each of our samples. And remember, each of our samples has a different barcode on them, so then you can pool our samples and then submit them for sequencing. And so using Pineapple Pi to process our results, you can see if you sequence your original plasmid library, Philip showed these plots before, these Lorentz curves. In an ideal world, you have this dotted red line that shows that you have a perfect distribution of the guide RNAs. And then what's important to show here is that when you do have toxin selection, you see the shift to the right. So we're having very strong selection, and you have a lot lower representation of the overall guide RNA pool, which is what we were going for with toxin selection. And so you can also look at these scatter plots where on the y-axis we have, for instance, cells treated with diphtheria toxin versus our control PBS samples. And everything in green is significantly enriched. And what was nice to see is the top 3 guide RNAs found were for HBEGF, which is the known receptor for this toxin. So if you knock this gene out, you get resistance to this toxin. So that was a nice experiment to see. We also found other genes involved in the diphthemide bus, the diphtheria toxin killing pathway. And another nice feature of pineapple pie is you can generate these plots that show, if you take, for instance, the top 100 guide RNAs from your enrichment list that is spit out on the other end of pineapple pie, you can look at the clustering of these guide RNAs between different samples. So everything in green is a high enrichment value versus red where you have a low read count. And you can see a nice overlap between these two toxins that both require heparan sulfate on the cell surface. And then if you look up over here, you can see that these 2 plant lectins that bind to other types of glycans each have their own subset of genes that are enriched when you treat with toxin. And so this is just a nice visual way to show over many samples what kind of guide RNAs are present and how they cluster with other samples. And so you can look at another scatter plot. This is for our gene eosaporin. Keep in mind, this is a small molecule that binds specifically to heparan sulfate and can carry a toxin, saporin, into these cells. And what's nice is we get enrichment of multiple genes. For instance, the #1 hit is a gene that's involved in heparan sulfate biosynthesis. So when you're doing these types of positive selection screens, it's nice to see if you're lucky enough to have genes that should be present. It's nice to look for these and see if they're enriched. But obviously what we're interested in are what are these other hits, these other guides that are present in terms of novel genes that might be regulating this pathway. And so as an additional screening method, we also designed a FACS sorting screen. And so what's nice about this screen is that we're not looking at toxins attached to the cell surface and getting shuttled into the cell. Here we're looking at just binding at the cell surface. So we can do this experiment on ice or at 4 degrees and look at binding of this heparan sulfate dependent ligand attached to a dye. And we can look for what cells, when a certain guide RNA is present, show decreased binding of this heparan sulfate dependent ligand on the cell surface. And so what we can do is we can then sort normal binding cells, quote unquote, versus these cells that have decreased binding at the cell surface. And so you can sort both these cells, then you extract the genomic DNA and run through the similar protocol with PCR 1, 2, and prepare for sequencing. And you can see here, if you look at the low binding cells versus the normal binding, you see a nice separation here of enriched guide RNAs versus our control samples. So you have this fold enrichment that's increased in the low binding fraction. And here we find multiple guide RNAs as well. They're involved in heparan sulfate biosynthesis, along with a lot of unknown genes. And so once you get these sometimes massive guide RNA lists, and this was touched on by Philip a little bit as well, is a nice thing to do is to you can look at gene ontology or pathway analysis, and I included a couple different tools that are free to use on the web, such as Metascape, where you can put in, for instance, your top 100 guide RNAs from your screen and look at which pathways are enriched. And the #1 pathway that is enriched is heparan sulfate biosynthesis, which is really nice to see in terms of validating our screening method. But as I've mentioned, what we're most interested in is how do we find new candidates, and how do we know which hits to go after. And so as a starting method, we have all these guides that were enriched. We introduced a very stringent method in order to choose which genes to go after. And so the first thing we said is that there had to be at least 2 guide RNAs present. And I think this is really important because when you're doing a transduction of your library, if there are 2 guide RNAs present in one cell, for instance, that will come along with it will enrich with another gene. So that's what's also nice with having 6 guide RNAs per gene, is that you can look, are there separate guide RNAs that target different parts of gene enriched in your sample? And We also wanted to look at least 10-fold enrichment of at least one guide RNA, and that guide RNA also had to have a low read count in our control sample. And we also looked at overlap between our different screens and different replicates. And so there's these 5 genes that we wanted to look at further, and they have, they could vary from transcription factors to growth factor signaling pathways. And so these were enriched in both of our screens. They had at least 2 guide RNAs and had very high enrichment. And so when you go in and validate or individually knock out these genes, we can do, for instance, a FAX binding assay, repeating this FAX sorting assay that we had. And we see decreased binding of this heparan sulfate dependent ligand when you knock out these genes that we found from our screen. So with that, I guess I already kind of mentioned all of this. Just wanted to thank my boss, Jeff Eska, and also Philip Spahn, who helped with designing Pineapple Pie and has helped with a lot of the experimental aspects of the project as well. And Nathan Lewis, undergrad Jing Lee, and the IGM Corps for sequencing, and the NIH for funding. So I'll take any questions if we have time. So thanks.
Questions for Ryan? I really I really like your sorting assay, like the low binding versus high normal binding cells. I don't think I've seen that before. It's pretty proper to the biology you're interrogating here, but it's absolutely a very nice signal you got there from those low binding cells.
Yeah, I know. Sure.
It was more a comment than a question. All right, so thank you, Ryan.